The Retort Season 1 presents expert views on the BTWC and CWC and the issues they face. This project is the product of a collaboration between Professor Lijun Shang, Professor Malcolm Dando and Dr. Brett Edwards. This initiative has been funded by the London Metropolitan University Rescaling Fund 2021, which was awarded to Professor Lijun Shang. We are grateful for this support.
Hello and welcome to this series of talks on the subject of chemical, biological weapon disarmament and non-proliferation. In this series, we will be hearing from a wide range of experts as we look towards the two major review conferences which will be taking place in coming months. In each talk, you will hear from a respected expert who will outline key opportunities and challenges in making pragmatic and holistic progress on this issue globally. This project has been kindly supported by the London Metropolitan University.
Hello, and welcome to this episode. With gain-of-function research back in the news, what better time to review the key principles and practices of this issue area? In this episode, we're very lucky to have a world-recognised expert speaking to us, so sit back and enjoy.
I am Nariyoshi Shinomiya, President of the National Defense Medical College Japan. I'm going to talk about so-called gain-of-function experiments or GOF study in this part. Life science and technology is rapidly developing, and concerns associated with technology in terms of biological weapons convention are changing as time goes on. In 1970s, traditional pathogens such as highly pathogenic bacteria, viruses, and toxins were the primary targets of prohibition. In 1990s, recombinant DNA technology made it possible to manipulate the pathogenicity of traditional pathogens. Rapid development of life science technology has dramatically increased the potential for the production of new types of biological weapons. Since 2000, there has been a strong focus on the development of advanced biological weapons using cutting-edge life science technology. Among those, the technological fields that have attracted the most attention are synthetic biology, reverse genetics, and genome editing. The proliferation of the Internet has made it difficult to control over such intangible goods. Let's see the relationship between technology and research activities in some specific fields. This slide shows trends in emerging life science technologies based on the number of published papers on PubMed website, which include high throughput sequencing, synthetic biology, genome editing, and pathogenicity modulation. The appearance of DNA synthesizer in 1986. Next generation sequencing in 2000. Cost reduction of DNA synthesis is around 2006. And the discovery of a new genome editing technology, CRISPR-Cas9, in 2012. Those epoch-making events dramatically stimulated research activities in each science field. Among recent life science technologies, reverse genetics made it possible to design influenza viruses at will. In 2005, a paper was published describing the successful revival of the Spanish flu virus. The genetic information of the virus was carefully read from pathology samples of the time, reconstituted into segmental plasmids, and then 12 of these plasmids were transfected at once into cultured cells. This created the same conditions as if the cells had been infected with the influenza virus, and infectious Spanish flu virus was recovered. However, this study was subjected to biosecurity review by the NSABP. a note added in proof at the end of the published paper stated that the experimenters were taking antiviral medication, that biosafety procedures were strictly followed, and that the purpose of the research was to contribute to public health. It was the influenza studies by Dr. Kawaoka and Dr. Fouché that brought gain-of-function research to the public's attention. As many of you know, controversial flu studies attracted people's attention in November 2011. At that time, there was no system to obtain international pre-approval for the activity related to dual-use research of concern. Most of countries did not have formal mechanisms in place to review the GOF research before starting. The Fink Report. The official title of the report is Biotechnology Research in an Age of Terrorism, issued in 2004, clearly presented 7 experiment classes as a way of thinking about dual-use research in the field of biotechnology research. As all of these 7 issues are related to the deterioration of bioweapons, and the influenza research in particular was considered to be related to be the 5th, pathogen host modification, which has come to be referred to as GOF research by those involved. The Fink Report is also known for its recommendation to create a new framework, the National Science Advisory Board for Biosecurity, NSABB, to check dual-use research of concern issues and to strengthen biosecurity policy. Let's take a look at how those avian influenza studies came to be conducted, and the NSABB's reaction as a reviewing body regarding dual-use research of concern. At the time, enhanced detection and preparedness for new strains of highly pathogenic avian influenza was an important public health issue. Also, research on the infectivity of the H5N1 virus was funded by the NIH. On the other hand, the NSABB's position was as follows. A summary of new findings from the research should be published. However, the detailed methodology for creating infectious viruses should not be included in the paper. The purpose of the research, public health benefits, and safety and security should be clearly stated. Next year, in 2012, a moratorium was proposed by scientists involved in the influenza GOF research. At the same time, they insisted on the importance and urgency of testing animal transmission of influenza. In February, the WHO held a meeting on this issue and concluded that it was important from a public health perspective. In March, the NSABB had a face-to-face interview with Dr. Kawaoka and Dr. Fouché. As a result of the review, all 18 NSABB members was in favor of publication of the Kawaoka paper, while the split decision for the Fouché paper was 12-6. In conclusion, both papers were approved for publication by a majority vote. The NSABB then presented its recommendation to the editorial boards of Nature and Science for acceptance of the papers for publication. Then the papers were accepted for publication in June 2012. To explain the Fouché's paper, this study modified the highly pathogenic avian influenza virus H5N1, which only infects birds, so that it can be droplet transmitted to ferrets. Moreover, they showed that the virus, so modified, shows 5 characteristic genetic mutations. This was a situation that raised concerns about misuse and abuse, since it demonstrated the means of intentionally changing the host range of the influenza virus from birds to mammals. The Kawaoka's paper used a less risky technique for infection control than the Foucher's paper. That is, they started with the H1N1 virus, which is less virulent than the H5N1 avian influenza, and replaced the hemagglutinin H1 portion of the virus with the avian H5. This led to the clarification of the structure of H5 that makes it more susceptible to infection in ferrets. But the virulence of the resulting virus itself was in a safer range than that of the highly pathogenic avian influenza H5N1. However, even after both papers were published, the debate continued, dividing virologists and many stakeholders in two opposite sides. In an attempt to break the deadlock, a workshop was held at the NIH in December 2012 titled Gain-of-Function Research on Highly Pathogenic Avian Influenza H5N1 Virus, an international consultative workshop. The main discussion points of the workshop were as listed here. Identification of current status of highly pathogenic avian influenza H5N1 gain-of-function experiments. Identification of research objectives and current status of research implementation. Significance of the research. Concept of research funding. Current framework. And what type of research should be conducted in the future. Following the results of a late 2012 conference entitled Gain-of-Function Research on Highly Pathogenic Avian Influenza H5N1 Viruses, Patterson et al. Presented draft criteria for research funding. The 7 items presented to Sciences Policy Forum the following year, 2013, presented a new framework. The debate over the research framework regarding GOF research seemed to have subsided. However, 2 incidents in 2014, the improper handling of anthrax bacteria at the CDC and the discovery of smallpox vials at the NIH, triggered biosecurity concerns about infectious disease research, and safety assurance for infectious disease research once again became an issue. In response, there were several developments in the United States. One was the strengthening of the pathogen control system related to dual-use research of concern by the US government. This added pathogens such as highly pathogenic avian influenza viruses and the 1918 reconstructed Spanish flu virus to the control list. The other point was that on October 17, 2014, the US government suspended research funding for GOF research and urged a reconsideration of its research policy. It also asked the NSABB to consider recommending new policies by the end of 2015. GOF research here represents the experiment using potentially pandemic pathogens, such as influenza, MERS, and SARS viruses, to change the infectivity or host ranges by manipulating their genomes. Pathogens conducted through such experiments are called enhanced PPP. The NSABB, which was tasked by the US government to study the ideal science policy for GOF research, discussed from multiple perspectives along the supported timeline. It also commissioned a risk-benefit analysis from Griffon Scientific, and received some 1,000-page analysis report at the end of 2015. Then, 2016, they convened a conference and, with the help of National Academy of Sciences, held workshops and other events. Finally, in May 2016, the NSABB published the final report titled Recommendations for the Evaluation and Oversight of Proposed Gain-of-Function Research. This is one of the statements within that NSABB report, Summary of Findings. Findings consist of 7 important items as shown here. 1. There are many types of GEOF studies, and not all of them have the same level of risks. Only a small subset of GEOF research has risks. 2. Risk can be managed and oversight of GEOF research of concern could be carried out properly if the policies are implemented effectively. 3. Oversight policies vary in scope and applicability. 4. An adaptive policy approach is a desirable way. 5. Possibly, some of GEOF research of concern should not be conducted. In that point, legal, ethical, public health, and societal values are important and need to be taken into account. 6. Both federal and institutional oversight, awareness, and compliance are required. 7. Many issues are international in nature. Also, the NSABB showed 7 recommendations. 1. GEOF research should be appropriately reviewed before accepting for funding. 2. A transparent and publicly engaged advisory board should be used. 3. The U.S. government should pursue an adaptive policy approach. 4. The oversight mechanisms should be incorporated into existing policy frameworks. 5. The GEOF research should be subject to oversight regardless of funding source. 6. Laboratory biosafety and biosecurity as well as awareness rating should be strengthened. 7. The engagement with the international community is important. The graphs show the number of articles in PubMed using GEOF research as a keyword. GEOF research in total declined once in 2017, but there has been a continued upward trend year by year. In addition, the number of searches for words combination with influenza, SARS, and potentially pandemic pathogen is not large but continues to be steady. Producing enhanced potentially pandemic pathogens about SARS or SARS coronavirus 2 may have been performed in the past, which may increase biosafety, biosecurity risks in the lab. So, we should now reconsider the nature of GEOF research that will generate EPPP, again after the COVID-19 experience. Recent reports supported this idea. Rewinding back a bit in time, the Inter-Academy Panel issued a statement on biosecurity in 2005 in response to accidents and bioterrorism incidents involving pathogens. In it, it quoted François Rabelais' words, "Knowledge without conscience is simply the ruin of the soul," and emphasized the 5 points listed here: awareness, safety and security, education and information, accountability, and oversight. GEOF studies that produce enhanced potentially pandemic pathogens must be reviewed from this perspective. These are references and further readings. So if you need more information, please refer to these papers or web pages. Thank you very much for your attention.